G protein coupled receptors and their effectors. GPCRs are a huge family of receptors that contain 7 transmembrane alpha helices. These receptors translate the binding of extracellular signaling molecules into the activation of GTP binding proteins. G protein coupled receptors are so named because it interacts with G proteins in the plasma membrane. Now, let us first understand that what actually G proteins means. G proteins are the specialized proteins with the ability to bind the nucleotides, that is GTP or GDP. G proteins are heterotrimeric proteins that consist of 3 different polypeptide subunits, that is alpha subunit, beta subunit and a gamma subunit. A G protein is said to be in its activated state when the alpha subunit of the G protein binds to GTP molecule, and it is said to be in switched off state when the alpha subunit of the molecule binds to GDP. Now, let us begin with the steps that how signaling takes place via these G protein coupled receptors and how the effector molecules gets activated. A ligand goes and binds to the receptor, forming a receptor G protein complex. All the receptors contains a ligand binding site for the binding of their effective ligands. The interaction with the receptor induces a conformational change in the alpha subunit of G protein, causing the release of GDP followed by the binding of GTP. In its GTP-bound conformation, the G alpha subunit has low affinity for G beta and gamma subunits, leading to its dissociation to form the complex. Each dissociated G alpha subunit is free to activate an effector protein such as adenylyl cyclase. In this case, the activation of the effector leads to the production of a secondary messenger. That is, the effector molecule activates the adenylyl cyclase enzyme, which leads to the conversion of ATP to cyclic AMP. These secondary messengers then in turn activate one or more cellular signaling proteins. A G protein is said to be on when its alpha subunit is bound to GTP. The G alpha subunits can turn themselves off by hydrolysis of GTP to GDP and inorganic phosphate. This results in a conformational change, causing a decrease in the affinity for the effector and an increase in the affinity for the beta and gamma subunits. Thus, following the hydrolysis of GTP, the G alpha subunits will dissociate from the effector and reassociate with the beta and gamma subunit to reform the inactive heterotrimeric G protein. Now, how the termination of the response takes place. Since the ligand binding results in the receptor activation and the activated receptors turn on the G proteins and the G proteins can turn on the effectors. To prevent overstimulation, receptors have to be blocked from continuing to activate the G proteins. This process is termed as desensitization, the process that blocks active receptors from turning on additional proteins. It occurs via a two-step reaction, out of which the first is the cytoplasmic domain of the activated GPCR is phosphorylated by a specific type of kinase, which is termed as G protein coupled receptor kinase, which is mainly a serine threonine protein kinase. Now, the phosphorylation of the GPCR sets the stage for the second step in the binding of proteins called as arrestins, which bind to the receptors and compete for the binding with the heterotrimeric G proteins. Via these steps, the secondary messengers produce in turn activate different signaling protein molecules. If you like my video, please subscribe my channel for more videos like, comment and share. If you have any queries, you can comment below. Thank you.